Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 1.6, nucleic acids. Today's focus is being able to describe the structure and function of DNA and RNA, especially how they encode genetic sequence and talking about the monomers of DNA, such as nucleotides, as well as the parts of the nucleotides, and also the overall structure of DNA and how that affects its function. And the suggested skill is Visual Representations 2A, describe characteristics of a biological concept, process, or model represented visually. Here we go, monomers and polymers of nucleic acids. And remember, our monomers are going to be like our building blocks, and the polymer is what is going to be built out of those building blocks. And there are two kinds of nucleic acids, DNA and RNA. And these are formed from the monomer nucleotides. So nucleotides covalently bond to each other in single strands, and multiple strands attach together through hydrogen bonds. So if we take a look here at this strand of DNA, or I guess double strand of DNA, this DNA helix, we can see that each nucleotide right over here, and we'll talk about the components of nucleotides in just a second, each nucleotide is covalently bound to other nucleotides forming the strand, and then these two strands are bound together through hydrogen bonds right over here. And again, you can see up here that when we're looking at nucleic acids, we're looking at CHOMP. And so that means that nucleic acids are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. And those are the essential elements within nucleic acids. Now, looking in depth into a nucleotide, which is again our monomer of the nucleic acid, nucleotides consist of three parts: a 5-carbon sugar, pentose, pent as like pentagon, 5-sided, and ose, sugar. And the pentose depends on the nucleic acid. So DNA and RNA have different 5-carbon sugar. And we'll look at the differences between DNA and RNA in a few slides. But then there's also going to be a phosphate group, and then a nitrogenous base. And this nitrogenous base is obviously based on nitrogen, but it can be an adenine, cytosine, guanine, thymine, or uracil molecule. And so the phosphate and the pentose sugar are going to form the backbone, and they're bonded covalently, just like we talked about in the previous slide. So as you can see here within each nucleotide that makes up the DNA strand, here's our 5-carbon sugar, here's our phosphate group, and here's our nitrogenous base that's going to be bound to the nitrogenous base of the other strand through a hydrogen bond. Now, when we talk about DNA strands as a whole, it's important to note that DNA strands run in the direction from the 5' carbon to the 3' carbon. That's their directionality. And when I talk about the number of the carbon, that means starting from the oxygen atom within the 5-carbon sugar, what carbon atom are we at? So as you can see here, we can look here, we see our oxygen atom that goes 1, 2, 3, 4, 5 carbon. And then here we have 1, 2, and 3. So this DNA strand is going to run from this 5 carbon here to this 3 carbon there. And both strands are going to run, both strands of DNA within the double helix are going to run 5 carbon to 3 carbon, but they are anti-parallel, meaning they're parallel but they're in opposite directions, which you can see here. This one is going to run in this direction. And the DNA is a double helix, and like we talked about, the strands are held together by hydrogen bonds between the two strands. And when we talked about the nitrogenous bases, it's very important to remember that they are certain base pairing rules. Adenine only pairs to thymine, and cytosine only pairs to guanine. A common saying to remember this is apple tree, car garage. And so the hydrogen bonds between these two base pairs are going to be what allow genetic encoding, replication of DNA, and also keeping the strands together. And the nitrogenous bases can either be 2-ringed, like adenine and guanine, or 1-ringed, like thymine, cytosine, and uracil, which is part of RNA. We'll talk about that in a second. And the 2-ring bases are known as purines, while the 1-ring bases are known as pyrimidines. It's kind of funny because the larger bases have the shorter title. And the role of nucleic acids is to store and express genes. So DNA is going to store all of your genetic information, and the various types of RNA are going to help in expressing that genetic information. So let's talk about RNA now. What is RNA? So RNA stands for ribonucleic acid, whereas DNA stands for deoxyribonucleic acid, and it has jobs related to DNA to code for proteins from the genetic information. So DNA is going to store your genes, and RNA is going to help express those genes by coding for the production of proteins. And we'll talk about proteins in the next video. So the three kinds of RNA, we have mRNA, which is what's going to carry transcribed DNA to ribosomes to make proteins. And, if some of these words sound unfamiliar, that's okay, we'll get to them in further units. But essentially, mRNA is our messenger RNA. It's going to carry a message of DNA. tRNA is our translation RNA, and that's the RNA that's going to translate the messenger RNA into amino acids, into proteins. And lastly, we have our RNA, ribosomal RNA, which is going to be a major component of ribosomes. And we'll talk about what ribosomes are in the second unit. And the nucleotides of RNA also contain a sugar and nitrogenous base in the phosphate group. But the sugar is different, the nitrogenous bases are slightly different, and RNA is only a single strand, which makes it smaller and it's easier for it to be replicated. So now let's compare DNA and RNA. So comparing DNA and RNA. DNA is going to have two strands, like we talked about. It's a double helix, and the two strands are anti-parallel. One's running in this direction, the other in that direction. And the pentose sugar is going to be called a deoxyribose sugar, meaning it's going to be missing an oxygen atom. It's deoxy. And the base pairings are going to be apple tree, car garage, ATCG, adenine, thymine, cytosine, guanine. Now RNA, on the other hand, is a single strand. It also runs 5-carbon to 3-carbon, but it's single strand. The sugar is going to be ribose. It's not missing an oxygen. We can look down here. Here's our 5-carbon sugar missing the oxygen atom right here, deoxyribose, and here's ribose. And then the bases are going to be AUCG. So instead of adenine binding to thymine, adenine is going to be binding to the base of uracil, which changes from DNA to RNA. But cytosine and guanine are still bound together. So these are the three main differences between DNA and RNA, and probably the most important ones are the number of strands and also the base pairing, because that's going to affect the transcription and translation of genes, turning this genetic information into something that can be expressed outwardly. So visually we can see, obviously we're going from 2 strands to 1 strand, and instead of having thymine that can bind to adenine, as we can see in DNA here, we see this adenine and thymine bound together. That's the base pair bound together with hydrogen bonds. We see uracil instead. So there you have it. Make sure you know your structure of DNA and RNA, your nucleotides and the components of those nucleotides, and the nitrogenous bases. Their function of storing genetic information and then expressing it. And of course, the differences between DNA and RNA. We have our 2-to-1 strands, we have our different sugar, and we also have adenine to, or thymine to uracil, different nitrogenous base. We'll see you in 1.7, proteins.